Today, our objective is fermentation and what are the features of a fermenter. Fermentation is the process in which there is incomplete breakdown of glucose. Fermenters are used for the fermentation of microbes. It is used for the large-scale, rapid production of microorganisms and their products. Fermenters may be designed for anaerobic or aerobic processes. Now, let's take a look at the features of an industrial fermenter. So, I saw a video from YouTube which is about fermenters too. I'll drop the link at the end of the video. Now let's watch the stolen video.
This in here is the medium of the fermenter, and in here you will have microbes, you will have nutrients in, and you'll have air going in. In particular, you'll need O2 going in most cases. The microbes that go in here are going to be ones that you consider particularly valuable. You want to grow lots of them. Maybe, for example, they are transgenic bacteria to make human insulin, in which case we're going to need it to be aerobic, and we're going to want to have lots of glucose in here, and we're going to have lots of amino acids in here. You're going to need these and other nutrients besides, because you want glucose for your bacteria to have fuel for respiration, and you want amino acids so that they can make proteins and grow, but also so that they can make human insulin itself. That itself is a protein, and amino acids are the building blocks of proteins. All this around here is a fluid, is a liquid suspension and solution of these various things. Now, of course, microbes themselves don't dissolve in water, and so given the opportunity, they will just settle out to the bottom down here. And that's no good at all, because the ones on the top, well, maybe they'd be all right, but the ones in the middle, well, they wouldn't get any amino acids or glucose, or indeed oxygen, which is going to be dissolved in this water. And you'd also get a build-up of carbon dioxide in the middle of those piles. So we don't want settling. Settling is no good for us, and that's why we have these paddle stirrers here to keep it going around, to keep it well oxygenated, and to stop CO2 from building up, and to make sure all microbes get a fair supply of food. CO2 itself will of course be made, and so that CO2 will come out here, and we have our gas outlet to allow that to come out. So we've got O2 going in, we've got CO2 coming out. In fact, we kind of need to do the gas exchange for the microbes in this way. How do we know when to do that? Well, we have these probes. They will detect levels of CO2 and of O2, and that will tell the computer what it needs to do. That CO2, of course, is produced by aerobic respiration. Aerobic respiration also generates heat energy. Now, we want the fermenter to be at a reasonable temperature in order for the bacterial enzymes to be working at their optimum temperature. But we don't want it to get too hot, and therefore typically actually we have cooling water going in. This is a cooling jacket here, and it will come out slightly warmer. It will be taking heat energy away, and so therefore it will cool the fermenter, keeping it at an optimum temperature, again as controlled by the computer. We want a tap for the product, and then you'll need to purify your product later on in downstream processing. And there's one little thing that maybe we've missed out on this diagram, and that would be here. That would be a filter for the air going in. It's going to be really important that we stop other microbes getting in. We have made, in this fermenter, a lovely, warm, gooey, salty, sugary, amino acidy, oxygenated solution, which is perfect for microbes to grow. Now, of course, that's not just perfect for our transgenic bacteria to grow, as we want them to, but it's also perfect for any other microbe to grow. And so we got to make sure those things don't get in there. So we filter for the air in, and that will keep the conditions sterile. That's really important. And of course, beforehand, you're going to want to clean it properly so that there aren't any bacteria in there before you start, and you do that with superheated steam. Thank you, superheated steam.
So now let's review what we have learned so far. Number 1. Water jacket. It's to maintain the optimum temperature so that enzymes can work at their best. 2. Air filter is required so that no pathogen or unwanted microbes can enter the fermenters. 3. Stirrers. They are used to mix all the ingredients properly and to inhibit settling. 4. Growth medium provides all the nutrient required by the microorganisms in fermenters. And last is number 5. Superheated steam is used to kill all pathogens. That's all. I hope this video was informative for you guys and hope you enjoy.